what did you put? What did you put? Yeah, no, it's right. Yeah, it's right.
I'm going to speak since it's dead. Just want to remind you that immunoglobulin is composed of 4 proteins that come together by disulfide bonds. 2 light chain proteins, 2 heavy chain proteins. The light chain and the heavy chain proteins in the variable end composed of where you bind an antigen. The heavy chain portion of the protein also has this very, very long constant region. This, in many textbooks, is also called the FC region. The light chain regions have a variable domain, a joining domain, and a constant domain, whereas the heavy chains have a variable domain, a diversity domain, so they have an extra variable rank domain, plus a joining domain, plus the constant domain. So I want to see if that's mostly what you saw here. Again, the antigen binds somewhere over here. These basically are bilaterally symmetrical molecules. The same antigen that binds here is the same antigen that binds there. Right? These 2 proteins are no different from these 2 proteins. It's a tetramer, held together by disulfide bonds. Okay, so the heavy chain gene which is really the important one. The light chain just follows everything the heavy chain does. The heavy chain gene has over 300 V segments, has about 20 V segments, has 6 J segments, and then it has, so that's all this is the variable domain, and this is the constant domain, but this constant domain here actually comes in several different versions. It comes in a mu version, a delta version, several gammas, an alpha, etc., an epsilon version. The mu version is the first one used, and I'll go over that in a little bit more detail. But everything starts with the functionality of the joining here together by gene rearrangement, followed by the usage of the mu constant region. That is the critical one that drives the functionality of this gene. So, just to remind you all, because I think there are some critical steps that you need to remember. Every V chain has a leader segment, followed by an intron, followed by the actual variable domain of the protein. So this is one V segment. Let's call it V1. It also, every variable domain has its own transcription start site. So that means that everyone has its own minimal promoter. Yeah? It's all varied. So I'll get to it in a second. So notice there is no enhancer associated with this V segment. And there is no enhancer associated with any of the 300 V segments. These all just have a minimal promoter, and that's it. Followed by a little part of an exon, which makes the leader sequence, which I told you gets cut off once it gets secreted to the membrane as it goes through the Golgi. This is essentially the de facto N-terminus of the protein, that you eventually use when you're creating your antibody diversity. So you have roughly 300 of these units. So that was, again, what the slide shows here. Right, you have 300 of those units. Next, further down in the genome, you have an array of these segments. Again, 20 of them. Each exon represents a different diversity domain. Again, a little further down, you have an array of J segments. Now, I noticed actually in the book, I don't think this way, and I have a feeling it's going to confuse you no matter how I describe it. I think the definition of an intron. So the definition of an intron, and I've never taught it this way before, so let's hope I don't confuse you. The definition of an intron is that which gets removed in RNA. Right, so when we talk about making a primary transcript, if you give a random gene like this, you make a primary transcript, you splice out the intronic sequences. Correct? So that's how these are defined as introns, because what ends up in the RNA are the exons. A little bit different, and I think your book treats it a little bit differently. Because these and these splice together and splice out this region, and these, the J's, splice out intervening regions, these intervening regions are not called introns. They're called intervening regions. They are essentially never intronic because they never end up in RNA. It's a sort of, it's hard to swallow.
Well, you say they're not intronic because they never end up in RNA. Yeah. So, okay.
Right? So, again, I'm not trying to confuse you. I'm just trying to make this a little bit easier to digest. Yeah. So that's what we described last week, and we'll go over again. If you D regions splice together, your first step in immunoglobulin rearrangement is D's spliced to J's based on these sequences called RSSs. This intervening sequence gets circularized and deleted. DJ recombination happens on both chromosomes. Again, I won't test you on this, but I think it's interesting to know that it happens on both chromosomes simultaneously. Yeah?
Because the removal of those areas occurs in the DNA itself rather than as an mRNA transcription process, then it's not an intron.
Correct. I said this, I never really thought about it that way, but I think that's how your book treats it, so I'll teach it that way. It's a good way to learn it anyhow. It's appropriate. So you have two chromosomes. DJ recombination occurs on both chromosomes. It is not the rate-limiting step. Any D has joined to one particular J, any D has joined to one particular J. So this could be D1 with J5 and D2 with J3, right? You just delete it away. So the DJ event happens biallelically, both chromosomes. And then one of the chromosomes gets selected for VDJ recombination. And that mechanism is poorly understood, although I think there is some treatment in your book where one of the chromosomes splices out genomic DNA and creates a VDJ recombination, VDJ joining event on one chromosome. As I told you, each of these mechanisms, let's say V1, each of these mechanisms joins the two fragments and puts extra nucleotides inside. That means you never know whether this VD is in frame to this DJ. You never know if the whole thing is in the proper open reading frame. Yep.
For the DJs or the frame, is there no?
There is absolutely no prediction mechanism to know whether they're in frame. And so what happens is, you have a constant mu region here. If VDJ has happened on this first chromosome, you have an RNA made. And by the way, I should say, the RNA gets synthesized because you've now moved V next to D, and there happens to be, way down here, a mu enhancer. A heavy chain mu enhancer. And when you make the VDJ joining, now that mu enhancer is much, much closer, and it activates transcription. You transcribe your gene, and you test it to see if it works. How do you know it works? Because you make a complete open reading frame that creates a protein that can traffic to the membrane and attach to the membrane. And signal back to the cell success. Now, I'm going to add some bit of confusion here. The heavy chain happens first before the light chain. So it's heavy chain, then light chain. However, you need a light chain to get to the cell surface. How does the cell do that? Yep.
When you say happens first, you mean it's expressed?
So rearrangement happens in the heavy chain locus prior to the light chain locus. There are 4 alleles of the light chain locus, but none of them undergo any rearrangement, and thus none of them produce any of those light chain rearrangements. However, you cannot get this to the cell surface without a light chain made. Nope. You need to test before you can get light chain rearrangement.
It's not the final light chain, it's just a simple recombined light chain.
So you're close. There's a surrogate light chain. There's another gene dedicated in these early B cells. So I won't bore you with all the details. There's pre-B cells, there's pro-B cells, there's pro-something B cells. Finally, you get B cells. There are about 10 or 20 different steps to get to a B cell. This is one of the very earliest steps. And in this very early step, there's a surrogate light chain expressed. So that you can allow that heavy chain that's been rearranged to get to the cell membrane. Because you actually have to make what looks like a pretty functional receptor, a tetramer, to get to the membrane. And the mu form here, by the way, has a part of the protein that has a transmembrane domain in it. So it's membrane bound. Mu, membrane. Mu is M, membrane. It's the easiest way to remember it. Membrane bound form. So you need this light chain. If you have successfully achieved VDJ rearrangement, you go on to start recombining your light chains. And again, one at a time. One allele, if it's okay, it tracks to the cell surface with your heavy chain, and you test to see if it's made properly. You get a different signal.
So successful VDJ rearrangement means that the translated protein sticks in the cell membrane.
That is correct. So failure to have this first heavy chain happen allows the cell to undergo a second round of rearrangement. And thus you now, if this fails, now this V2 gets recombined. And you test, you make an RNA, and you test. If both fail, goodbye, cell is done. Yep.
Is this part of the self-test? Where it's testing to see if it binds itself?
Yes. Self-test has to do with T cells. And, it starts with the T cell self-tolerance, not with the B cells. I'm pretty sure. Autoimmunity is run through the T cells, not the B cells usually. We can talk after class. So, you have this rearrangement event. If you fail to get a mature protein, you test again, you make a new one, and if that one succeeds, then you go on to the light chains. And again, you go through them one at a time. Yep.
So, but if both parties of light chain fails, so the cell dies as well?
Yep. Well, you get 4 chances,
4 chances fail.
and then you're done.
All right.
But the surrogate light chains is from a different gene,
Yes.
so it's a different gene.
Yes.
The light chains that are.
Yes. It's just a regular protein that is made only in pre-B cells, because you wouldn't want to outcompete your heavy chain in mature B cells. Yep.
Is successful light chain expressed in just BJ joining?
There's a constant region too.
Well, yeah.
BJC.
Okay.
Yep. So, yeah.
You said 4 chances, not 2? Or 6?
Well, you have 2 alleles of lambda and 2 alleles of kappa.
Okay.
Although I have to tell you, like, I forget which one's either the lambda or kappa. I think not all cells go through all 4 rounds, but technically you have 4 chances. So when you take a full out immunology, you'll see it's even more complicated. So, and again, I said to you that this goes through the RSS sequences, which are the spacers between your V and D segments, or D and J. And again, this is in genomic DNA. So I'm sorry I might confuse you when I say we look at splice sites, we look at the DNA analog, but splicing happens in the RNA. Here, these are real DNA sequences. The effect is on the DNA sequences. The RSS targets are DNA sequences, not RNA sequences. And again, I showed you the video for this, and it's really quite cool. And again, this is what I showed you about a non-productive rearrangement triggers you another shot. Again, I showed you this end joining, which adds extra nucleotides, so you get this cut here, and then through TDP, you start adding in extra nucleotides, and you get randomness. Remember, for the protein to be in frame, you have a 1 out of 3 shot at each of the end joining to get you back in a proper reading frame. That doesn't even mean that you're going to make a good protein. Maybe you make a protein that poorly traffics to the membrane. You're done too. Just because you made a full-length protein doesn't mean you successfully transported it outside of the cell. It's a little bit of a nuance, never tested on. So this is again what I showed you. Here are the splicing events. These are this is the membrane-bound form here that initially gets made. Eventually, you now undergo what's called class recombination, where you switch from the membrane-bound form to one of the secretary forms. And therefore, that secretary form does not have the transmembrane domain. So here are the many types of different IG molecules you get, the mu, the delta, the gamma, and each one has various roles. And the triggers for them, actually, I'm not even 100% sure. I think it's poorly understood how you trigger class recombination. So class recombination is the following. It's a different mechanism completely from RAG1 and RAG2. It involves a whole set of other proteins. And here, class switching undergoes, for example, here, this is class switching where you've deleted CMU, delta, and gamma 3. And you've just expressed gamma 1. So this is where you recombine between different of the constant regions. Class switching involves two other proteins. You involve AID, which is and non-homologous end joining pathways. I won't test you on it, but what I want you to know is that there are other recombination mechanisms that exist in the cell in order to create DNA deletions. We touched upon this in the last 5 minutes. I'm going to go back to something I touched upon in one of the earlier lectures. Once we understood that DNA rearrangement was possible, and basically enzymes that cleave DNA at recognizable sites, people started looking very carefully to see whether we could identify any enzymes in particular target sequences. And what came out of that was a protein called pre-recombinase and a target site called LOXP. And the second site, and actually more of them are coming out nowadays, flipase, flip recombinase, and its target site called FRT. LOXP sites and FRT sites are both 34 nucleotides. They both contain 13 nucleotides, anti-parallel, I'm just drawing one strand, plus 8 specific nucleotides in the middle. So this is true for both LOXP and FRT, this sort of structure. This is specific, these are inverted duplications. What that provides you with is the following. If I have DNA here and I incorporate a 34 base pair sequence that is LOXP, let's say between an exon that I'm interested in getting rid of, let's say exon 3 of a particular gene, and now I overexpress Cre recombinase, I can now delete out everything between those LOXP sites. So a very specific example I will give you is the following. And by the way, just so that you realize that, if this is one LOXP site, this is the other LOXP site, and actually let's draw it like this, with red. When you make this deletion, you're getting half of it comes from one side, half comes from the other side, everything in the middle is gone. Yep.
Is it are LOXP sites palindromic? Or are they
Except for the 8 nucleotides.
Right.
Right.
And they're both going the same direction.
Right, so they have orientation. I won't go into orientation, but they do have orientation, and they do need to be in the same orientation. What happens if we do this? Just curiosity to see what the class says. What happens if you have this? They invert. So nothing gets deleted, but it gets inverted. So this causes an inversion. You won't be tested on that. So if you have a sequence like this that's 3 in this orientation, when you invert it, it'll be like this, inverted. So what this gives you, for example, is, let's say I had 4 cells, and I Cre recombinase expressed in this, and let's call it the left leg. And I wanted to, so I used my enhancer trap lines or whatever lines to get Cre expressed only in the left leg. This is the right leg. I can ask, what happens if I delete my gene in the left leg? while keeping it normal on the right leg. For example, it gives you an opportunity to ask, what are specific deletions in specific types of cells going to give me, what is the phenotype I'll observe if I control my pre-recombinase in a particular cell? So remember, all 4 cells are normal for this gene X, let's say. This is X wild type, X wild type. Now this is X deletion, X wild type. And you can ask, this is your right leg. So in the same animal, you can ask, what is the effect? And why would you want to do this is because the following reason, deletion, X deletion over X deletion is death. Because that gene is involved in just setting up embryos. So you can't even look. You can only look later on in adult animals or later on when the animal has survived. This may be a little bit too nuanced. Are there questions about this? I'm going to stop right here. Yeah?
Do we need to know this?
You will eventually need to know free recombination and lox P deletion. Yes?
I don't understand how you express it in one leg.
Because you have a transgene that's expressed in one leg